Hello and welcome. Today I'm going to be going through the FRQ questions for the AP exam from 2023. We're going to go over each question individually and we're going to go through how to approach these questions as well as how to answer them. So we're starting off with this first question here, and this is one of the bigger questions that comes at the beginning. This one I believe is worth 9 marks, and you're going to get 2 of these really large types of questions. So as you can see, this one has quite a lot of writing in it, and it has both a diagram as well as a chart. So there's a lot of information here. I'm going to read through it. We're going to break it down as we go. But these are the types of questions you can expect to see where there's, a lot of information. Okay, so let's start off by just going through and reading it. So it says, in eukaryotic microorganisms, the PHO signaling pathway regulates the expression of certain genes. These genes, I'm going to call them faux target genes, encode proteins involved in regulating phosphate homeostasis. When the level of extracellular inorganic phosphate is high, a transcriptional activator known as FO4 is phosphorylated by a complex of 2 proteins, FO80 and FO85. As a result, the faux target genes are not expressed. When the level of extracellular PI is low, the activity of the FO80, FO85 complex is inhibited by another protein known as FO81. This enables FO4 to induce the expression of these target genes. A simplified model is shown in figure 1. Okay, so this is just explaining to you the pathways, and that's in writing. I prefer looking at the visual diagram. So this didn't tell us anything that the diagram is not telling us. So I'm going to go through and kind of break down this diagram. So it's showing you A versus B, and these are 2 different types of conditions. So A is showing you a high phosphate environment, so when you have loss of phosphate available. B is showing you a low phosphate environment when there's lower phosphate available. So when there's high phosphate, we see that we have our FO4 protein, and FO80 and FO85, these ones are connected to each other like a dimer. They're going to result in the phosphorylation of FO4. Okay, so FO4 gets phosphorylated, and as a result, this is going to be inhibited. So this is our DNA and our faux target genes. It mentions in the first part of the question that when FO4 is phosphorylated by a complex, which was our FO80 and FO85, the target genes are not expressed. So you actually get no expression here. This phosphorylation of FO4 basically makes it inactive, and as a result, you're not expressing any of those faux genes. And we're going to get to it, but it obviously makes a little bit of sense. If you have high phosphate, you might want to lower the amount of expression that you have. It's in a feedback loop. So I'm going to just write here that this is basically inactive due to the phosphorylation. Okay, so with high phosphate, you're going to get no expression of your target genes. If we have a low phosphate environment, we're going to see that FO81 is actually going to inhibit FO80 and FO85. That's what this little line means. When you see a line and then there's a little cross at the bottom, this usually means inhibited. Either way, it tells you in the question that the activity of FO80 and FO85 is inhibited by FO81 when there is low PI. So FO81 is inhibiting FO80 and FO85, unlike what's happening here where it's active. And then as a result, you're not going to get FO4 activation, right? Because you can see here that you need FO80 and FO85 to cause FO4 activation. Since we're inhibiting it, we're not going to get FO4 phosphorylation. As a result, since the phosphorylation makes it inactive, this FO4 is not phosphorylated, so it's actually going to be active now. And then as a result, you're going to start to express your faux target genes. So they're going to be expressed. And then as a result, you're going to get your faux target genes, mRNA, and then eventually the proteins. That's just going to be our pathway. So this is the breakdown of our schematic right here. I wouldn't worry about it too much for right now. We're going to keep going with the rest of the question. So this says that to study the role of different proteins in the FO pathway, researchers used a wild-type strain of yeast to create a strain with a mutant form of FO81 and a strain with a mutant form of FO4. Okay, so we have a mutant FO81 and a mutant FO4. So I'm just going to highlight where those are in the diagram because those are going to be the most important ones. So we have FO81, which is our first one in the pathway. And then we had a mutant FO4. So those are these over here. Okay. In each of these mutant strains, researchers measured the activity of a particular enzyme, APAs, which removes phosphates from its substrates and is encoded by FO1, a FO target gene in table 1, which we'll see. They then determined the level of FO1 mRNA relative to that of the wild-type yeast strain, which was set to 10. Okay, so the one thing that I wanted to point out here that I feel like a lot of students would have struggled with is this portion here, where it's telling you what metric they're using to measure the expression. So ultimately, we're trying to figure out whether we're mutating, like what the effects of mutating FO81 and FO4 is on our FO target gene expression. Okay, so we're going to be mutating what's going on over here, and we're seeing how that affects the expression. Now, they're telling you that the target gene they're studying is known as FO1. Okay, so that's the gene, and it's either going to be not expressed or expressed, okay, depending on our mutant. So now we're going to look at this table here and break this down. So this table is APA's activity and relative amounts of FO1 mRNA in wild-type and mutant strains of yeast in high and low phosphate environments. Okay, so they give you the strain, and there's 3 different types. The wild type, whenever you see wild type, it just means like the normal one with no mutations in it. FO81 mutant, which they tell you means that the FO81 is not functional, and then FO4 mutant, where it's a non-functional FO4. So if we look at the measurements up here, you have APA's activity in a high phosphate environment. So again, APA's activity is our metric for how we're measuring gene expression. So APA's activity in a high environment versus a low environment of phosphate. And then you also have relative amounts of FO1 mRNA in high versus low. So again, FO1 is going to be the gene that we are expressing. It's ultimately going to affect APA's activity. So both of these are really looking at our level of expression for high phosphate environment versus low phosphate environment for each one of these yeast strains, okay? So I know this was a lot, and I'm trying to go through and explain everything, including in this question. The questions are not too difficult for this one, despite how, complex this looks. So we're gonna go into the questions and break them down. We're gonna start off with A. And A is a couple parts. It says, describe the effect that the addition of charged phosphate group can have on a protein that would cause the protein to become inactive. And then there's a second part. We'll just start with this first part here. Okay, so a lot of the times these ones will start off with more so like theoretical questions that you should know and don't really have much to do with, the actual parameters of this. It's asking you, the addition of a phosphate group, why does that cause a protein to be inactive? We talk about this in, I believe, Unit 4 when we do cell signaling, and the idea that the attaching of a phosphate group can turn proteins on and off. So they act like a molecular switch. They're asking you, why would this happen? Okay, so for this one, the correct answer is going to be that by adding a charged phosphate group, think of it, it's charged, it's a large group, there's lots of electronegativity. What's going to happen to that what's going to happen to the protein is it's going to change shape. Okay, so for this one, I'm going to say that that charged phosphate is going to result in a change of shape. Let me just get my pen. Okay. So we have a change in shape, or you could say structure, And we know that structure affects function, right? So that phosphate group is being attached, it's affecting the shape of the protein and then ultimately affecting the function of it, which is why, it's becoming inactive. Okay, so that's a pretty theoretical question. So the second part of this question here is asking, explain how a signal can be amplified during signal transduction in a pathway such as the Faux signaling pathway. So it's asking how it can be amplified, and that means the signal to be increased, right? You want to spread that signal, you want to increase the strength of that signal in a signal transduction pathway like this. How are you going to do that? We talk about amplification in a signal transduction pathway. That's one of the steps, right? You have reception, amplification, response. So the way that you're going to be able to amplify a signal is by having your protein or your signal interact with a bunch of proteins. So what that looks like is, okay, I have this signal protein. It's going to activate these 3, right? And then these ones are going to go on to activate 3. So this is how you amplify a signal. You have one molecule interact with several proteins, okay? So what that looks like in writing is, we'll just say each enzyme interacts with multiple, we can say proteins or targets. Okay, so that's how you're going to increase the strength of that signal. Each one of your enzymes is actually going to act on, for example, 5 enzymes rather than it's a 1-to-1 ratio, and that way you're going to increase your signal. Okay, so for B, it's addressing the table. So this says, based on table 1, identify a dependent variable in the researcher's experiment. Okay, there's more to this, but we'll start off with that first question. So again, this is just going to come down to, your ability to read these tables and understanding what an independent and a dependent variable is. So your dependent variable is always going to be what's being measured. The independent is what you're changing, the dependent is what's being measured. So in this case, you kind of have several options for here because there's multiple columns. You can say that it's either, and they only, I believe, identify a dependent variable. So they're only looking for one, but there are 2. It is APA's activity and amounts of faux mRNA. Okay, so those are what we're measuring, therefore those are our dependent variables. So you can say APA's activity, or, and again, you don't have to say both of them, just one of them is fine, relative faux 1 mRNA. Okay. So the second part of this says, justify the researchers using the wild type strain for the creation of mutant strains. This is a very common question that they'll give you as to, we're interested in, what happens when you have a non-functional faux 81 and non-functional faux 4. So it makes sense why we have these strains here. They're going to ask you, why do we also have this wild type strain? The purpose of having the wild type there is to use as a comparison, okay? You're trying to see the effect that faux 81 mutation or faux 4, doesn't matter, has on the overall, expression in these yeast cells. You don't have anything to compare that to unless you run the exact same experiment using non-mutated yeast, okay? So you always have to have a reference to go back to, right? Because I could tell you, oh, it's going to be, 0.4 APA's activity when I knock out faux 81, but compared to what? Is it upregulated or is it downregulated? I don't know because I don't know what the standard is. So that's the purpose of the wild type. You're going to be comparing both of these back to your wild type, okay? So in terms of justifying this, it's so that you can see the difference in terms of the experimental results between the strains, okay, that are caused just by the mutation. Okay. So to observe differences an expression. And we'll say between the strains due to or as a result of the mutation. And then you can kind of, I would add a little extra here that just says, you know, compare to wild type for reference, or as like a standard. You always need to have these, okay? Alrighty, now the last part of part B says justify the researchers using mutant strains in which only a single copy of the pathway was mutated in each strain. Okay, so again, we're still looking at experimental design for these. They're asking you why you have a FO81 mutant and a FO4 mutant. Why do you not just have one strain that's a mutant in both of them, right? Because you can make a FO81 and FO4 mutant, you know, both of them being mutated within the same yeast cell, but why do we not want to do that? Instead, we've opted to do the mutations separately. So the reason that we are not doing them combined and we're doing them separately is so that we can see the effect of each individual mutation, right? So if I have a mutant that has a mutated FO81 and FO4, and you know, I observe that it's upregulated. Well, I don't know what mutation is doing that. I don't know what protein is responsible for that. It could be FO81 or it could be FO4. You can't tell what each of those mutations are doing if they're combined. When you do these, you only ever want to change one variable, because then whatever happens in your dependent variable in your results is a direct response to the one thing that you changed, right? So the reason that you're going to want to do this is you want to be able to test the mutations separately and determine what's responsible for what. Okay, so test mutations separately. Or you could also say observe which mutation is responsible for, you know, whatever will happen. Okay, so this one was a little bit more complex, but if I were to, you know, give you kind of a reference, if I wanted to see, you know, the way that temperature affects an enzyme, I'm not going to change temperature and concentration or temperature and pH, because if the enzyme is, you know, not functional anymore, how do I know that's because of temperature? It could also be because of, you know, pH, because I changed both. You only ever want to change one thing so that you can say for certain that the change in my dependent variable was due to this one change that I made, being in this case FO81 or FO4 mutant. And you're comparing that, again, like we said, to the wild type. You're never going to compare FO4 to FO81. You're comparing it to the wild type to see that difference between your standard. Alrighty, so now for C, I'm just gonna get rid of this to make more room. C says, based on the data in table 1, identify the yeast strain and growth conditions that lead to the highest relative amount of faux mRNA. Okay, so get rid of this. C is asking you to identify the strain and the growth conditions leading to the highest relative amount of FO1 mRNA. So we're only looking, in this case then, at these two columns, right? It's not asking you about APA's activity, it's only asking you about FO1 mRNA. So we're looking for the highest relative amount. So within here, now we're just gonna look for which one has the highest amount. 0.1, 0.7, 0.6, 0.3, 0.9, and then this one's 10. So this is the highest number out of all of these. They're asking you what the strain is and what the condition is. The strain for this one is the wild type. And what's the condition? Low PI. Right? Because we're looking at high phosphate versus low phosphate. our low phosphate environment for our wild type is going to give us our highest expression. Okay. The next part of the question says, calculate the percent change in APase activity in wild type yeast cells in a high PI environment compared to that of wild type cells in a low PI environment. Okay. So let me erase this. Now we're looking at APase activity. So they're asking you for percent change, and there is an equation for this that you should know going into your exam. So APase activity in wild type, so we're actually only looking at wild type, in high PI compared to low PI environment. So we're actually only looking at these two numbers, because they're asking you specifically for wild type and then APase activity, and we're going to do percent change. So percent change is typically final minus initial over initial times 100%. Okay, so this is the equation for percent change, and for this one, percent change is a little bit complicated, and they're giving you two options to do these, because in the case of this experiment, there really isn't an initial and then a final. When you do initial and final, it's, you know, before treatment versus after treatment. In these cases, you're not taking the same yeast, putting it in a high PI environment and then putting it in a low PI environment. These are separate yeast. They would be flasks or whatever. So you don't technically have a final versus initial. You have two separate measurements. Hopefully that makes sense. So in this case, you can't really tell which one is the final and the initial. So they actually accepted two answers. So for this, you could do 17.3 minus 0.5 over 0.5. So that's, you know, saying that the final is 17.3 and the initial is 0.5. And you do times 100%, which I believe should give you 3,360%. Or they also accepted this answer as well, because it's not really a final versus initial. They're two separate measurements and two separate, you know, yeast experiments. You could have also done where 0.5 was your final and 17.3 was your initial. So it doesn't matter which one you took as your final or initial, they would have accepted the answers. So that one would look like 0.5 minus 17.3 over 17.3 times 100%. That's going to give you negative 97%. So either one of these would have worked. Okay, make sure before going into your exam that you know your percent change equation. I don't know if it's given on your formula sheet, but still, you should know it. Okay, so now finally, we have part D. So we actually haven't done really anything with our complex pathway yet. So the pathway looks really overwhelming, but all of these questions have been, you know, kind of about this chart. So I think D is going to be our final one. It's a couple parts. It says, in a follow-up experiment, researchers created a strain of yeast with a mutation that resulted in a non-functional FO85 protein. Okay. Based on figure 1, predict the effects of this mutation on FO1 expression in the mutant strain in a high PI environment, and then provide reasoning to justify. Okay, so FO85 is what we're now changing. So we're going to go to our schematic here, and I'm going to highlight where FO85 is in the pathway. So FO85 is going to form a dimer with FO80, and ultimately it's either going to phosphorylate or not phosphorylate the FO4. And as a result, that's going to affect gene expression. However, in this one, they're asking you to predict the mutation in a mutant strain in a high PI environment, okay? So they're asking you specifically for high phosphate. So we're actually going to ignore the low phosphate one, because we're only focused on, okay, I have a mutant FO85 in a high phosphate environment. What's going to happen? So if we look here, Fo4 is phosphorylated by Fo85 and Fo80. If Fo85 is mutated, basically this is not going to work anymore. If Fo85 is non-functional, you're not going to get your phosphorylation, right? Your Fo85 is doing the phosphorylation. If it's inactive and it's not working, you're not going to get a phosphorylated Fo4. So instead, you're going to remain as an unphosphorylated Fo4, okay? And as a result, we saw that a phosphorylated Fo4 meant no expression, and we saw in this one that a unphosphorylated Fo4 results in expression, okay? So I'm going to say that again. These two diagrams showed us that when Fo4 is phosphorylated, you get no expression. When Fo4 is not phosphorylated, you do get expression. And if we mutate Fo85, you are no longer able to phosphorylate Fo4. All of your Fo4 is going to exist in the unphosphorylated form. So you should actually get an increase in expression, okay? You're not going to have any phosphorylated Fo4. As a result, you're going to get increased expression. So it's predict the effects of the mutation on Fo1 expression. So as a result, Fo1 expression will increase. And they're asking you to justify it. So the reason Fo1 expression is going to increase is because Fo4 can't be phosphorylated anymore. So Fo4 is not phosphorylated, and then I'll just write due to mutant, is it Fo85? Yeah, due to mutant Fo85. And so now that it's not phosphorylated anymore, it's going to remain active. So Fo4 is now active. To drive expression. This is a lot more than you have to write, but I just want to make sure this is clear because in my opinion, this is kind of the hardest one in the set. Okay, so this is how you answer this one. In terms of what they accepted, you could say that, so you had to say that the Fo1 target genes are going to be expressed, and that is because there is a non-functional Fo85 that's not going to phosphorylate, okay? Again, as we saw, phosphorylation inhibits. When it's not phosphorylated, it is going to drive expression. With a faulty Fo85, you're not going to get any phosphorylation. You're always going to have it in its unphosphorylated form. So then you're going to drive expression. Okay, so that one was a big one. That's question 1 for these FRQs. I believe, yeah, it's 9 points total. So you can mark how you did these in terms of the breakdown. I believe this one is 2, B is 3, this one's 2, and I believe this one is 2. Okay, so you can mark your mark yourself with those. Typically, these underlined ones that I have here, or the bolded, indicate how many points that it's worth, because these are key terms. You need to know the difference between identify and justify. Okay, so that's question 1.